Good morning students, welcome back to our zoology class. Hope you are doing everything well and good. Now let us go on our today's class. Fine. So we are discussing chapter 6, evolution, right? And our today's topic is biological evolution. So let us go on to our class. So in the previous sessions we discussed about the origin of life as well as this geological time scale, right? So in the origin of life we discussed about these various theories which is explained by this various evolutionists regarding this origin of life, right? And under this geological time scale we discussed about this age of this earth. So the age of this earth is divided as various eras and it is subdivided as these periods as well as epochs, right? And of course this flora and the fauna were found during this period also. So everything was tabulated as one tabular column as a geological time scale, right? So those things we have discussed in the previous sessions, and let us continue our today's session. Fine. So in this session we are going to discuss about this concept regarding this formation of protobions or probionts, and one more thing is regarding this one of this experiment which is conducted by the persons Urey as well as Miller, right? So formation of protobions as well as this Urey-Miller experiment we are supposed to discuss by today's class. Fine. And let us see what are the things is given under this title, right? So the figure which is displayed there is protobions, right? And let us see what is protobions and how it is formed, right? So in the previous classes we discussed, so the biotic things are evolved from this abiotic substances, right? So from here also under this biological evolution we are going to discuss how these biotic things are evolved from the abiotic things, right? So and in this evolution is already they informed in the primitive year there was some abiotic substances. So from the abiotic substances the biotic substances are created or evolved, right? So that's what, so this process has happened step by step gradually, okay? So the first stage only here it is known as protobions. So how these protobions are formed we can see now, right? So abiotically produced molecules can spontaneously self-assemble into droplets that encloses a watery solution and maintain a chemical environment different from their surrounding. Scientists call these spheres as protobions, right? So the abiotically produced molecules is gradually started to assemble like a droplets and it is maintaining one structure. It is different from their environment, right? So this spear shaped structure has name as a protobions by the scientist, right? These protobions, actually the word it is proto, the previous, bions is related to this biotic things. But this is not the actual cell. The first cell is not yet formed during this period, but this is a previous stage of this cell we can say. It's almost like, right? So hence it is known as a protobions, right? And let us see what are the stage, changes or modifications may happen in these protobions to produce as one living cells. Fine. So the figure we displayed there is lipid bilayer sheet. So the bilayer sheet is mentioned there. So bilayer means it is two layer is there. So this sheet actually it is present in this membrane of the cell, right? And let us see how is it formed. So as we said in this previous term, that is protobions. So these protobions started to assemble as the solution which is present inside, okay? So these protobions started to assemble this one of the substance which is present inside. So that substance is known as liposomes. So liposome is nothing else, it is known as lipids, right? So the liposomes are lipids in a solution that can self-assemble into a lipid bilayer, right? So these protobions consist of this liposome that will self-assemble as two layers. So that is known as a lipid bilayers. Okay, actually if you look at this ultrastructure of this cell membrane, that is a plasma membrane, we could see this lipid bilayers, right? So this is this beginning stage, how this membrane consists of this lipid bilayers, right? So the protobions, consist of these liposomes which is assembled to form as a bilayers. So bilayer is a structure which is present in the plasma membrane, right? Okay, so that's what we could say. Gradually the changes has occurred, right? And let us go on this the next stage now. So this figure which displayed is conservates. So in the previous class even we discussed about this conservates, right? So conservates means large colloidal particles that precipitate out in aqueous medium, or the first pre-cells which gradually transformed into living cells. So conservates are the first pre-cells. So cells. So now here we are giving the term as a pre-cells. So this is a previous stage of the cells, right? But it do not consist of this substances like as actual cell. It is a previous form of this cell. So that is known as a conservates, right? So the conservates consist of this nucleoprotein and some nutrients inside, right? So and this is producing one limiting surface membrane around itself. Have you got? So these conservates consist of this nucleoprotein as well as the nutrients. So this is producing one limiting surface membrane around itself. So when it is producing this surface membrane, these characters look like or similar like this viruses, they said, right? So or it's similar like a free living genes. It's like a genes or it's like a virus, they said, right? So when the conservates, so now the conservates form is becoming like a virus, not virus, like virus. Its character is similar to virus. Have you understood? Okay, so now we can go for the next stage again. So the figure now we displayed is known as a protovirus, right? So now we say the conservates became like a virus. Now the next stage what they said is, so the genes, actually this conservates started to behave like a virus. The character is similar to virus, or they say it is like a free living genes, right? So now these genes, the group of genes united to form like one protovirus. So this also not a virus now, it's a protovirus, means like a previous stage of this virus, right? So the conservates behaving like a virus, or it's like us behaving like this, free living genes. So these genes are united together to form a protocells or protovirus. Okay, so these protoviruses are now, it is somewhat similar like our present virus. So protovirus has the similar characters like our present virus, right? Okay, so and they said two major cell types that appeared during this time were significant. So by this time there is two types of the cell type were available, that is one is Monera, another one is Protista. So after this virus stage there is two types of the cells, they said Monera type as well as this Protista type, right? So let us differentiate this, what is Monera as well as this Protista. So this figure we can say as Monera. So Monera in the sense actually it is the earliest cell. It contains clumps of nuclear protein embedded in the cell substance. So in the cell substance, the nuclear protein is clumped there, right? So they are considered as an ancestor to this modern bacteria. Actually, so Monera form, they say the nuclear protein is, embedded in the cell membrane. Along with the cell membrane, it is embedded. Okay, so this look like Monera, but Monera is this ancestor for modern bacteria as well as the blue-green algae, right? So now let us see what is, just this figure for this bacteria as well as this blue-green algae. Okay. So now we displayed about this Protista. So this difference they have noticed here for Monera and the Protista is, in Monera the nuclear protein is embedded in the cell membrane, right? And moreover, Monera is this ancestor for modern bacteria as well as the blue-green algae, right? But in case of this Protista, they said, and nuclear protein clumps that condense into the central mass surrounded by the thin membrane. So the nuclear protein is condensed in the central mass. It is surrounded by the thin membrane. Hence, this membrane is separating this nuclear protein from the cellular substance. But this nature is not there for this Monera. In the Monera, we say the nuclear protein is embedded in the cell membrane. But in case of this Protista, nuclear protein is surrounded in the center, which is separated by this thin membrane. So the membrane is there between this nuclear protein as well as this cellular materials, right? So that is this Monera as well as this Protista. Fine. Okay. Then now this Monera form as well as the Protista form is created. Then what happened is, there was this demand of these food substances in the nature. So when this population started to increase, there was this demand of food, or this source of food is reduced or declined. These Monera and this Protista, ancestors Monera and the Protista form, started to adapt themselves to with the different food behaviors. Different mode of this food behavior is there, like we could say parasitism, saprophytism, predator or animalism, and chemosynthesis or photosynthesis, right? So what do you mean this parasitism? Parasitism in the sense, one organism completely depend another one organism for its food, shelter, and so on, everything, right? And the second type is saprophytism. In this case, an organism used to get its nutrients from this dead materials. That is known as saprophytism, right? And the third method is predator or animalism. In this case, one animal used to kill this another animal for its food. So that's what it is, using the another animal as its prey, right? So that we can say as predator or animalism. Fine. And the next one is chemosynthesis or photosynthesis. So in this case, this especially the plants or doing this process like a chemosynthesis or photosynthesis. So of course you may know about this photosynthesis process very well. So during this process, this lot of oxygen will be eliminated, right? Let us look into this photosynthesis. So during photosynthesis or chemosynthesis, the plants used to absorb this light and the carbon dioxide as well as the water. So of course during this process, this oxygen is eliminated into this atmosphere. So when this photosynthetic organisms number is increased, of course automatically this oxygen synthesis also increase in the atmosphere, right? So first there was no oxygen in the environment. We said there is methane, ammonia, hydrogen as well as water vapor were available. Now the oxygen also synthesized. Okay, so how the oxygen is synthesized? Of course we have to trace it back step by step. We have to go back and in which way this oxygen is evolved, right? So by this photosynthesis process, how the photosynthesis processes begin? Of course when this demand of this food is increased in the environment because of the production of this Monera as well as Protista. Understood? So since this oxygen level is increased, then this condition is little bit changed, right? So already we said in the primitive atmosphere, ammonia as well as this methane is available. So this oxygen, it will combine with this methane as well as this ammonia, and it will produce this carbon dioxide, nitrogen as well as this water, right? So now let us look at this equation for this. So in this equation, the atmospheric oxygen combined with this methane, and it is producing this carbon dioxide as well as water, right? Meanwhile, those oxygen, atmospheric oxygen may combine with the ammonia or two, then it will produce this nitrogen as well as this water. Okay, so when this oxygen level is increased, of course it will combine this ammonia and this methane in the atmosphere, and it is producing this carbon dioxide, nitrogen as well as water, right? So since this oxygen level is more, of course it is enhancing this aerobic respiration. So what do you mean aerobic respiration? So the respiration with oxygen is known as aerobic respiration. The respiration without oxygen is known as anaerobic respiration, right? So here if there is an amount of oxygen is increased, it is enhancing this aerobic respiration. So here the foodstuffs are producing this energy, right? And finally because of this, prokaryotes as well as this eukaryotes are evolved, right? So the overall concept here is, from this abiotic things the biotic things are evolved, right? So this concept begin from this biotically produced molecules is going on for a series of reaction, and finally it is produced this biotic things like prokaryotes as well as eukaryotes, right? So that is all about this biological evolution. So based on this concept, other two persons known as Urey as well as Miller, they have explained the same concept with this one experiment, right? And let us see this experiment which is explained by these persons. Fine. So this is the experimental setup which is made by this Urey and Miller in the laboratory, and their concept is biotic things are generated or evolved from this abiotic substances. So already in the theories of evolution we have said, of course, the Big Bang theory as well as this biological chemical evolution explains there was some gases which was present in this primitive earth, such as methane, ammonia, hydrogen as well as water vapor were present. So in this inorganic substances, if there is this lightning and this UV radiation, volcanic events may cause this energy there, and because of this it may undergo some series of reactions, and finally these inorganic substances may become as an organic substance, right? So this is a concept already we have explained in this theories, right? So as per the same concept only, Urey and Miller also conducted this experiment, and finally they have proved this concept also with this experiment, right? And let us see this how they conducted this experiment. Okay, so this is made as per this natural reaction. In the nature how the reaction takes place, as per that basis this setup is made, right? So first of all, one round flask like structure is there, round shaped flask. There this gases such as this ammonia, methane as well as hydrogen is filled, right? And so as like this environment, then this water vapor is passed inside from the bottom. So if you see, look at this bottom, the bottom another one flask is there water is filled and the water also boiled, right? So when the water is boiled, it is producing this water vapor. The water vapor will move upward and it will mix in these gases, right? And to produce this energy, they are using this two tungsten electrodes, and through this they are passing this electricity. So it is producing this energy. So this energy is sparked inside these gases. Okay. So here inside this flask, actually the gases were there. Water vapor also passed as well as the energy also passed there. Okay. So these may make this reactions inside, and finally it will produce this. The water vapor is moving downward. When it comes downward, it consists of this one condenser. So this condenser roll is, inside this condenser they are passing this cold water. When they pass this cold water inside, on the way actually water vapor is coming. So when this cold water mix in this water vapor, the water vapor will condense and it will become as a droplets, like a rain, right? So it is becoming as a droplets. So the droplets will move downward and finally it will be collected in the U-shaped tube. Then it is collected in the conical flask. Right? So the same experiment is conducted, continuously this is done around 1 week, right? So when finally they analyze this sample which is present in the conical flask, which is collected from this experiment, so when they analyze, they could find this, glycine, alanine, beta-alanine and aspartic acids in this sample, so which was not present in this atmosphere in the beginning. Right? So actually the atmosphere consists of methane, ammonia, hydrogen as well as water vapor only. But when this electricity is passed inside, of course it is producing these organic substances such as glycine, alanine, beta-alanine as well as aspartic acid. Right? Then later also they have conducted the same experiments and they could find this all the types of these amino acids, nitrogen base also they have noticed. Fine. So of course this experiment clearly explains this biotic things are organic substances or created from this inorganic substance, right? So the same experiment I have explained with one animation video, so that also will give this clear illustration regarding this experiment. Fine, let us go on this animation video.
In the 1920s, Oparin and Haldane hypothesized that the atmosphere of the primitive Earth contained gases such as nitrogen, ammonia, methane, carbon dioxide, water vapor, and hydrogen. Oxygen did not become a part of the atmosphere until the advent of photosynthesis millions of years later. It was further hypothesized that under these conditions, organic molecules could be formed if there were a source of energy such as electricity from storms or ultraviolet radiation from the sun. In 1953, Stanley Miller and Harold Urey tested this hypothesis. They constructed a closed system with a reservoir for water, sampling ports, a chamber for the input of electrical energy, and a condenser to convert the water vapor back to a liquid. The researchers then added water, hydrogen gas, methane, carbon dioxide, ammonia, and nitrogen. The water was heated to form a vapor, and electrical sparks were generated to mimic lightning. This apparatus simulated the possible conditions of the early Earth. When the water was analyzed after 1 week, they found simple carbon compounds such as formaldehyde and hydrogen cyanide. These compounds then combined to produce formic acid, urea, and amino acids. In similar experiments carried out over the next several years, various organic compounds have been synthesized. These experiments showed that simple organic compounds, including the building blocks of proteins and other macromolecules, could be formed from gases present in the primitive atmosphere with the input of electrical energy.
Hope you understood about this one. And from this we can give two assignments there. So the first one is explain this formation of these protobions, and the second one is explain Urey and Miller experiment. So the first one you can go for the first paragraph. Of course in that paragraph you could find this 2 mark, 1 mark also, and it will be asked as a 5 mark question also, right? And Urey and Miller experiment you have to draw, you have to give with this diagram, and experiments must be explained here, right? So hope you will do it in this at home and make it as an assignment and learn the portions and submit it on the day of this correction camp, right? So hope you will be doing it well and we are expecting it. So underline this, all these important key points here, and go through this, read it well and underline all these important areas and cover it up. Fine. So next class we can meet with this next title. Under that, please stay at home and do your work very well and perfectly. Fine.